awards for research, teaching, and service, including the NSF Career Award, the ASEE Chemical Engineering Division Award, as well as an award for innovation in computer-based chemical engineering education. Her research is funded by the NSF and NIH, amongst other. And so today she's going to talk about multiscale modeling for tissue remodeling damage and treatment. So Ashlee, it's a pleasure to have you, and the virtual floor is yours.
Thank you so much. All right, so thank you all for attending, and I appreciate those who will watch the recording in the future as well. So I really appreciate the opportunity to be able to connect with all of you, and I know that there's not this opportunity for in-person interaction, but I would appreciate questions at the end, and then feel free to follow up if you'd like to have any discussions about anything I talk about today. I would be happy to connect in that way. So my lab focuses on multiscale modeling, and most of the questions we look at involve problems of tissue remodeling. So this is an equilibrium process where our bodies, when healthy, are continually turning over the tissue in many of our organs. So you could think about your skin, you get dry skin, it sloughs off, your body replaces that skin with fresh, new, more resilient skin. That might change in aging, it might change in disease conditions, but that generally is happening throughout adulthood. We're interested in this balance between tissue formation and tissue degradation. You need to remove the old tissue and replace with new tissue. But in some types of diseases, this balance can be dysregulated. The two processes may still be happening, but one of them may be out competing the other and result in a net growth of tissue. So for example, in the context of cancer, where we have too much tissue formation, or in the context of some eye disorders where we have blood vessels that are forming, causing vision loss. And then we can have on the other extreme conditions where there's too much tissue loss. So for example, osteoporosis in aging, we have loss in bone tissue. We also in my lab investigating the onset of these conditions. So starting from a healthy balance where both of these processes are happening, what kind of stimuli tip the balance toward the onset and further progression of those diseases? And if you're in a disease state, what kinds of stimuli might get us back toward this more healthy equilibrium or steady state between those two processes? We look at pharmaceutical drugs, nutraceuticals, things that are derived from foods that we eat. We also look at the role of the immune system in these, whether that's a chronic inflammation due to maybe a comorbidity like diabetes, or the role of infections and other kind of toxins that may trigger immune responses as well. So this is a snapshot of a Zoom snapshot of my team. We still meet remotely as a computational lab, even 5 years post-pandemic here throughout the pandemic, we facilitate students working remotely on our team as well. So a couple of these students have graduated within the last few months, but I kept them in here as I'll showcase a few of the highlights from their work. And so without this team and our funding sources, we would not be able to do the work that we do. So when I talk about multiscale systems biomedicine, I use that biomedicine term explicitly rather than systems biology. A lot of systems biology focuses at the genetic level and intracellular. So what are the networks of cell signaling that might happen inside single cells? And while those are both very important, my focus is more on getting towards those clinically observable tissue level or whole body level types of things that can be measured and detected in biopsies and other things. So we sometimes leverage intracellular networks, but we're very interested in communities of cells and how they secrete various chemical factors, how they move around, multiply, the populations of them, and these interactions that are mostly at the tissue level. but tissues give rise to organ level function and dysfunction. And then we can think about therapeutics that are just delivered through different modalities, whether that's through the digestive system, the respiratory tract, through the circulation, directly into a target tissue, and then how that spreads and those drugs get delivered to the location of interest. We don't do a lot on the social network side, but that's also an important other extreme of the scales that one could consider as people might interact and spread, for example, COVID. So my team tends to focus on the cellular and organ level networks and dabbles in molecular networks and whole body types of dynamics. We focus on building models, and the types of techniques often are motivated from chemical kinetics. We think about, as a chemical engineer, this is definitely a key subject that we leverage. We've looked at the kinetics or the speeds or rates of change of biomolecules, cells, and even entire organisms. So we have some earlier work that deals with bees and how they respond to pesticides. And so this formalism can apply to any kind of entity that we could track through population or mass conservation balances. We're thinking about describing their reactions or interactions and how they might change states dynamically, generally through networks that involve chemical communications. We also look a lot at mass transport. So we think about biomaterials, so those might be inside the human body or an animal tissue, or they might be engineered biomaterials, things like tissue engineering, cell cultures, organoid related biomaterials, polymer drug delivery devices for extending release of different therapeutics. And most of these materials have pores, have fibrous structures, have heterogeneities, either by design or because of stratifications within that tissue. They also might be experiencing reactions or disease conditions that might be driving changes that are dynamic and heterogeneous. So we're very interested in these complex microenvironments and then how that influences the movement of small molecules, larger molecules like proteins, but also cells and how they move through those biomaterials. And my lab is completely computational. We have a team of different collaborators for various experimental components that we work on, but I want to highlight today various computational methods that we use, and I've kind of organized the talk around showcasing different computational methods in my lab. So this is the spectrum of things that we're doing these days. So the base of our work highly comes from ordinary and partial differential equations. In chemical engineering, we often associate kinetics with ODEs and transport with PDEs. So that's where the basis of those formalisms come from. We do quite a lot with programming, of course, in Python and MATLAB, but making interfaces or apps or live scripts, these kind of tools, mostly to communicate our research results to collaborators or to the general public or for use in classrooms. We use some different open source tools and COMSOL also for fluid dynamics, biomechanics, different finite element solvers. My team is not, we're not experts in biomechanics and fluid dynamics. We're not generally generating new algorithms in those spaces, but we use those tools if they're appropriate for the problems that we're trying to study. We do work with dynamical systems and data analysis, data analytics, and we're moving more into that space as we have been partnering with experimental collaborators over the last several years to generate more and more data that then, in order to develop our mechanistic models, it would be helpful to have some data-driven insights to help us identify mechanisms and places to go in the next stages of our models. And we also have adopted agent-based modeling paradigms in the last several years. So these are such that we think about cells as discrete entities that have some rules that define their behaviors. We've also applied agent-based modeling in that context of the bees. So the bees were individual discrete agents. So there's not really a limit to what size scale you use. But in the formalisms we use for ABMs, we typically think about cells. as the discrete agents, and we use partial differential equations to describe small molecule movement and couple these PDEs to the agent-based rules for the cellular behaviors. We do different ones where we might think about the size or the shape of cells. So this particular screenshot is from a tool called Computational 3D, where each cell is composed of multiple pixels. I'll show you some results today from some work where they're more center-based methods, and so each cell is just a sphere and is not they don't have different sizes associated with them, and we don't care about the cell shape. We just care about the populations and interactions of those cells. And there are other agent-based formalisms that exist that sort of focus at different aspects, but I'll show you the fizzy cell-based, center-based techniques in a little bit. So these are a smattering of the different applications we've pursued in the last several years. So I will tell you a bit of our diabetic kidney disease story later today. I won't be able to spend any time talking about cancer metastasis and cell migration, but that's a very active area in my lab. I also won't have time to talk about our work related to nutritional supplements that we bring in through the digestive system. They trigger various immune responses that are protective and bone restorative. We have some really lovely preliminary results in this area, but they're unpublished at this time, and so I won't be able to share that story with you today. We do have a long history now of working on different immunotherapies, immunology types of applications. So we started this work in tuberculosis, and then we extended this to COVID when the pandemic started, and then we coupled that lung infection work to fibrosis that was consistent across these different organ systems and our tissue remodeling. So I will tell you about that work today, and I will tell you about some of our eye drug delivery work. So we organize the lab under these different organ systems: bone, lung, kidney, eye, and cancer. And again, for this audience, I want to highlight the different modalities of computational tools that we're using. So I'll try to focus there. You won't see a lot of tissue-specific conclusions in the work that I'm showing, but I want to kind of give you the highlights of some of the different tools that we use to answer different questions. So I'll first start with the lung. And so when the pandemic began, Paul Macklin, whose expertise is in multicellular modeling in cancer biology, he assembled a team to leverage the agent-based software that he had and some work related to cancer vaccines and those kind of aspects, and brought together people who had infectious disease experience, both experimental and modeling, people who had been using agent-based models, people who had done viral infection dynamics, several people who'd studied influenza exclusively or the related SARS and MERS infections. And so brought together this team, which we called the SARS-CoV-2 Tissue Simulation Coalition. And I want to give a big shout out to Mohammed Aminul Islam in my lab, where Aminul was a major driver for my lab's participation. As a postdoc, he really embraced this project and went quite far with it. The work has had several iterations on bioRxiv and has not yet been submitted to a journal, but is really quite a tour de force of resources that are in the multiple versions of this bioRxiv preprint. So we developed a lung tissue simulation. So the idea is when the virus comes in, thinking about the infection that happens at the ends of the lung airways in the alveoli. And the alveoli, these air sacs, have very thin epithelial cell layer where air exchange with the vasculature on the other side of the epithelium is facilitated by this very thin layer. And so we modeled that single cell layer thick of epithelial cells, and we started with some immune cells present and then added different rules related to an infection-induced immune response. So this is very much a multi-scale problem. So we're thinking about these very small viruses within this host tissue, and inside of a single cell, we are thinking about the dynamics of virus replication. So here the orange circles represent virions, the viral particles that can get into the cell through a series of dynamic processes represented by differential equations. They can replicate, infect the cell and change aspects of the cell, and then eventually be released from the cells. And these orange virions can then spread through the domain, and we use differential PDEs to describe their movement. So this viral binding and replication is at the intracellular level. And then at the tissue scale, we think about once this cell has been infected, it sends out some different cytokine or chemokine signals by secreting various factors that diffuse through the tissue environment. And there's a whole host of different immune cells and various signaling tissue level signaling communications that are encoded in this model, as well as communications with other tissues. So the lymph node is where a lot of immune cell replication and recruitment happens and then comes back into the target tissue. So we've included lymph node activation and the populations of these different cells that then come back into the tissue to respond to infection. And so one of the earlier versions of the model looked like this. So the blue background is the uninfected cells. You don't see the viral particles, but they are there and they're diffusing. And then you start to see the gray cells that have been infected. You have a whole host of these immune cells that have different kind of speeds and roles and interactions with each other. The red ones I'll point out are the CD8 plus T cells that are responsible for trying to kill off infected cells so that that infection can't spread further. And my team leverages these CD8 plus T cells and some rules with them. But what you'll notice is as this evolves over time, you start to see holes form in this tissue. And if we let this evolve, we'll eventually have depleted almost all of the tissue. So we explored different immune responses, different rates of different processes in the immune cell recruitment and the roles of different immune cells, but you get these really large portions of the tissue that are just decimated by this infection. And what's missing in this work is the wound healing part, that after we do kill off cells in our body that have been infected, the tissue gets restored through fibrotic type of mechanisms, and the body tries to turn off that acute infection and inflammation. So my part of the team, led by Aminal, sought to add that wound healing type of mechanism. So after an infected cell has recruited the CD8 plus T cells and these very inflammatory macrophages, we made a rule that says if they're both co-located, that means they're responding to that infection, then it will start to turn off that inflammatory signal and will instead transition the inflammatory sets of macrophages into an anti-inflammatory pro-fibrotic phenotype that will secrete various factors that lead toward, and we are considering TGF-beta as the main one of those, to recruit and activate fibroblasts that are already in the tissue and from other tissues that then come in and heal the wound. They're depositing more collagen. If that doesn't turn off, that can lead to extended fibrosis. And in our computational work, we explored the magnitudes of how much of this TGF-beta factor might be released from the mobile macrophages that are moving around the tissue, as well as when an infected cell dies, it can trigger the release of some TGF-beta that was deposited in the tissue already. And so we consider those to be stationary sources of latent TGF-beta, and this to be mobile sources of TGF-beta. So we explored the different influences of those types of things mechanistically. So by having this variation in the sources, we're considering these two different ways that the TGF-beta can be sourced and how that responds in fibroblast dynamics. And so we did some computational experiments where we only have the stationary sources and then we don't have the mobile sources. And what you see here is populations of the cells after a certain number of days. I think these are on the order of 10 days. And in the back, so all the circles are different cells. The blue are the original epithelial cells, and then all the other colors are different immune cells. But this color bar in the background, this is the collagen that has been deposited in response to the purple fibroblasts. And so if we have this very stationary source, there are higher pockets of collagen where there was an original infection. And so we have these very localized depositions. This is the area fraction of that collagen deposition. If we instead mechanistically switch to only allowing the mobile sources of the collagen, we see a much more diffuse pattern. We see a larger area fraction covered of cells or of pixels in the grid that have above a certain threshold of the collagen concentration as changed from baseline. We also then considered the combination of those sources and whether if one of them was high and the other had high or low. And basically what we got as the main result is that this mobile macrophages were very much driving this. These sources are dominant, that when these are high, you don't really see much of a difference between the stationary sources. Whether they're high or low, we get very similar outcomes here. And then similarly, when the macrophages are low, even with high and low values of the stationary sources, we get pretty similar dynamics. Now, at the time, we were dealing with statistics that were more like cell counts and these very rudimentary area fractions. My lab is now trying to explore things like topological data analysis and more sophisticated spatial statistics to better characterize the differences between these types of agent-based model results. But those were not things that were incorporated into these earlier studies. We also compared area fractions with different mechanisms turned on and off, and the clinical values were more in the 35% type of range. So we were showing, which ones needed to be on in order to approach that kind of clinical relevance. And we also did some other in silico experiments where we could accelerate or accentuate different facets of the model. And so things like having the stationary sources secrete or trigger more and more of that stationary source to be released from the tissue. And so we get these very high pockets of collagen due to this extended period. We could also consider the fibroblast uptake of that TGF beta and how that might change the dynamics of the system and the patterns that would emerge from that as well. So we could play with a lot of things mechanistically in this agent-based model type of paradigm. We extended this work to look at questions related to patient age and sex specific differences. And so we coupled our agent-based model to more of a whole body ODE-based model for the angiotensin signaling network. That's very common models used in pharmacokinetics of blood pressure regulation to control this hormone level. But two of the key receptors that are involved in catalyzing this reaction are influenced by SARS-CoV-2. And so the virus actually binds to receptors that are important in this angiotensin signaling process. So for patient age and sex related differences, we considered what the number of these receptors are available in these different virtual patient groups and propagated that through this network and through the fibrosis model. I won't go into the details of the results, but this is an example of coupling a cellular-based model, whole body sort of dynamic ODE kind of pharmacokinetics type of model and a tissue specific agent-based model paradigm. And this work just came out a few weeks ago. For the eye work, this is work in collaboration with Caitlin Swindle-Riley's lab at The Ohio State University. And on my team, Jabia Chowdhury and Eduardo Chasen Ruiz are the main trainees who've been working on this project over the last few years. And in this work, we're focused on the application of wet age-related macular degeneration. So in the back of the eye, there can be new blood vessel formation in this condition that causes degenerative vision loss and you have all of these neovascularization, and the treatment to be able to reach the back of the eye where there are these blood vessels is to monthly inject an anti-VEGF therapeutic into the vitreous to aim to the back of the eye. You can imagine this is painful. Patients don't like to comply with that 1 month frequency. They might shift to being more like every 2 or 3 months. They might not do it at all, and they would just rather lose their vision than to have things injected in their eye that frequently. So just big trade-offs there therapeutically for patients. So we've been looking at other ways to reduce this burden for patients. And in this first paper that we put together, Eduardo searched for different drug delivery devices and biomaterials applications that had been designed to target age-related macular degeneration. And he considered different mathematical approaches, whether those were mathematics for these different drug delivery materials or mathematics for the pharmacokinetics in the whole eye, aiming to target the back of the eye. So it's a really lovely, comprehensive review on both the modeling and experimental aspects of this problem. So what our collaborator has come up with is two different biomaterials approaches. One is more classic microspheres, where just 2 layers to extend the release lifetime. These are on the order of 13 microns, and I'll just note this is 10 times larger in height than that. They're just shown here illustratively, but these capsules are shown in this inset to be smaller than a grain of rice, and their diameter is such that they could fit inside of a hollow bore needle. One's normally used to inject the drug. Instead, if they could inject this capsule, the idea is to extend the release to 1 or 2 years rather than each month for just a solution-based injection. And these are composed of layers that are formed by electrospinning around a metal rod. So they spin them around like cotton candy, and then put a second layer on that's hard to penetrate or slows the release. And they remove this polymer from the rod, and then that interior is hollow, and they can load the entire interior with drug molecules, and then they cap the ends. And it's generally by heating the ends to melt them together. And so these rods then can have a large payload. They very much extend the release. The microspheres are much easier to model, but the microcapsules are more interesting and more relevant therapeutically. So we're using data information from both, from preliminary studies from their lab, but with the idea that we're really driving towards these microcaps or these capsules rather than the microspheres. So we are using combinations of different modeling modalities here. So generally for the microspheres and microcapsules, we have early work that was in MATLAB, so very much discretizing PDEs into symmetric, having only one spatial dimension, and then transforming that PDE with the method of lines into a discretized spatially, but then just using differential equation ODE solvers to solve the time-dependent part. So our bilayer spheres are actually quite easy to solve in this paradigm because they don't have, they're not biodegrading. The microcapsules have more interesting boundary conditions, have this hollow interior, so we're using COMSOL to solve them. But we also compare our results between MATLAB and COMSOL for the microspheres as just verification on our own part that we're applying the boundary conditions consistently between these two different modalities, especially between the 2 layers, the interfaces between these two materials. We also are using COMSOL and finite element methods more broadly to understand the dynamics within the eye. So shown our microsphere, our microcapsule, and our eye geometries. And these are not to scale. These are much, much smaller than the larger eye geometries. So for the drug release problem, the simplest modality is to consider diffusion only. And at a first pass, this should be relevant because these two biomaterials that we're using to comprise these layers are very, very slow to biodegrade. We're considering partition coefficients between those layers of material. We're using the data from the experiments in these materials to find diffusion coefficients and partition coefficient between the two layers. And when we consider the microspheres compared to the data, very well, very good fit for just considering diffusion and having the right geometric parameters of these specific microsphere combinations. So that works very straightforwardly. When we consider the capsules, these different formulations have different starting porosity, and that porosity comes from fabrication, not from replicas of the same, but from intentional design choices to make them more porous, to adjust that release rate. And so if our model only considers diffusion and only considers these geometric differences in these large capsules, that's not sufficient to describe and fit this data. And so earlier work that I had done as a PhD student was about degrading polymers that had evolving porosities that were reaction dependent. And so where you could switch from having small particles release their payload faster, when there was a reaction that could change the porosity of the material, that could enable large particles with large porosity to then release their entire payload faster than the smaller particles. So we want to incorporate some of that work into these, and then also consider some different things related to the hydration of these particles when there's manufacturing related pores, not biodegradation related pores. Those manufacturing pores are there from the onset, and there'll be those drug molecules that are available at the surface of the pores can move more easily than those that have to move through the polymer bulk. So we're trying to add those mechanistic pieces, but then also our collaborator is moving beyond polycaprolactone and chitosan to include polymers that do degrade. So this was polylactic co-glycolic acid. She's trying to explore this same polymer without the G. So just polylactic acid that is known to biodegrade, so that if you injected these might stay for years, and you don't want them to be empty but remaining. So there'll be some design trade-offs on having them last long enough that it's better than monthly injections, but not so long that they're empty and just sitting there. So we want them to be able to degrade. So we're working on that from an experimental as well as a modeling side. In the meantime, while we're waiting on new data from experimental standpoint, we also in parallel worked on the eye pharmacokinetics work. So we built these finite element models for the eye geometry, both in human and rabbits. Most of the data is coming from rabbits. We will be doing rabbit studies with those controlled release devices. For now, we did an injection bolus. So what's the volume that gets injected? And then after that immediate time point, then that bolus gets to spread out. But this is where we put the initial dose, and we consider different injection locations. We consider this top layer to be the boundary between the anterior, the front part of the eye, and the posterior, the back part of the eye. We always allow there to be a flux across this. It's very well known that there's fluid exchange between the front and the back of the eye. It's less well established about what's the right rate of flux from the back of the eye, or if there is any elimination. So we considered two boundary conditions there with PDEs, whether there's zero concentration or zero flux boundary condition. There was also some debate in the literature about whether there was a velocity imposed from the front to the back of the eye. And if it's diffusion only, we put velocity as 0, but we also explored a couple different convection considerations. And it seemed to be that those coming from mathematics and ophthalmology tended to say there was no velocity, and those coming from more of a biomechanics or a fluid dynamics background were imposing a convective term. So we wanted to be able to compare to data to see, which ones were they based on people's training and backgrounds and preconceived like which terms should be there, or based on real physiological information. And it's very hard to measure these in humans, so that's why there may be these debates. So we explored different boundary conditions and different convection cases. We did this in rabbits and human. We also considered, there's clinical guidance on what the angle should be of being able to inject this to not go through the middle of the eye and to not impair the vision. So we had this angle known, but we didn't know the depth. And so we put in different depths. And this is the rabbit scale and the human scale, but these are the same distances and the same volumes, just the rabbit is much smaller than the human. So it looks that the injection is it occupies a bigger fraction of that rabbit vitreous. So I'm only showing the human results here, but we did those simulations where we said we always had anterior elimination. We considered yes or no for the posterior elimination. So elimination at the fastest where we had the infinite sink boundary condition. And we considered whether we turned on convection or not. And the rabbit data is actually very different than the human data. The rabbit data is much more up here. The human data is much more consistent with these cases 1B and 2B. So if we look carefully, those are both cases that involve posterior elimination. So it didn't matter too much if convection was on or off. If you allow flux through the back of the eye, and this is as fast as possible, then that's very consistent with the human data. Now, there's limitations in the human data. It's very hard to do this experiment. So very little data in the literature. In a rabbit, you can sacrifice the animal and measure the concentration of the whole vitreous. It's time consuming and arduous, but you can do that. You can't do that in a human. These data came from when patients had small sections or small samples of their vitreous removed for other types of information to be gained or other kinds of treatments. And then it was, they could then measure this in that context, but they weren't, they're limited. I'll just say that. And so in the paper, we also do this we look at the log scale of this to make better comparisons at these values when it looks like it's approaching zero. So we have more resolution. But we compared the different dosing locations, these different boundary conditions. So we thought very much like, how can we interpret the data and the physiology based on using these PDE-based math models? So next things that we're doing, with the capsule, we can embed it into the eye. We can put it in different locations in the eye, and we'll need to, with the animal experiments, determine if this capsule moves around at all. We also are working right now with the microspheres data. I told you it's pretty easy to simulate that in MATLAB. We can get very quickly we can get a cumulative release profile from those microspheres. And so we've had some undergrads coupling that work with compartmental pharmacokinetics. So they might say, here's what happens in this region of the eye, the middle of the eye, the front of the eye, and not having a spatial component, but having these three different compartments and mapping those to data. So my students are working on, if we didn't use an injection, if we use these microspheres, what's the right design of the microspheres to keep the drug above some target threshold, reaching the back of the eye for as long as possible. So we're using those to think about design of experiments, but also to design rules for optimizing these drug delivery devices. So there's lots of future directions here, but enabled by ordinary differential equations and partial differential equations. The final system I'll talk about is our kidney-related work. And this is a long-standing team effort supported in my lab. And so Minu was the very first graduate student in my lab, and Michelle was the very first undergraduate student in my lab. And this has continued to be the subject of multiple students' PhD work, lots of undergraduate researchers, master's researchers as well. And so we're interested in damage to the kidneys due to diabetes. And we focus on multiple cells in this region called the glomerulus. So if you zoom in on the kidney, the nephrons have this filtration zone that has blood vessels that are covered in various layers of cells, and the blood gets filtered across these layers of cells and collected in this capsule and then is excreted into the urine. And so we're interested in the different cells and these kind of different zones of cell-cell interaction. So we can have the cells of the blood vessel, the endothelial cells interacting with the mesangial cells that provide structure to the space in between the blood vessels. We can have interactions between the endothelial cells, the basement membrane, and podocytes that wrap around the whole outside. We can have areas where there's all three cell types communicating. In disease, we can get these mesangial region can be very fibrotic that causes different biomechanics and pressure and things on the blood vessels and squishes them. So we're very interested in all of these kind of interactions, but we've been building these up cell type by cell type over several years now. So first, we're thinking about how hyperglycemia, this blood glucose level, damages the glomerular filtration barrier. And we started with podocyte cells. These are the cells that wrap on the outside. And this is looking at a view in the capillary lumen. The small molecules in a healthy case will move across this basement membrane and be able to move through the spaces between podocytes. But in disease conditions, the podocytes can peel off. That's changing the filters in between them. It changes aspects about the basement membrane. The podocytes can actually become detached, and you'll start to detect protein in the urine, and this is irreversible damage. So we're very interested in the onset of this and different chemical factors that could slow this process. And just really trying to understand this multicellular both transport and reaction type of processes here. So we started with building biochemical ordinary differential equation-based networks for the interactions in different cells in that environment, how they respond to glucose, which is the green is representing, how they can respond to different therapeutics. We also have done similar things now with macrophages and endothelial cells, and I'll tell you a bit about that work. We've thought about tissue structure. So our first approach was maybe we could couple this in an agent-based model. We have some more elaborate techniques now. And we also were considering the intercellular transport using PDEs to couple what's happening at the cellular level to the whole tissue level. So Haryana was a PhD student in my lab who graduated in the summer, and he took multiple approaches to model the renal fibrosis in the mesangial tissue. And so things related to, if a cell was in a different porous environment, how would its signaling distances be affected by fibrosis? He also had more of a this is an ordinary differential equation-based model where he put in different processes and different cell types that would regulate fibrosis and used experimental data in the literature to parameterize this model, and then thought about different ways he could perturb the system that could have pharmacological types of actions. And so we published this in Frontiers in Pharmacology in the fall, but it's a really well thought out paper from Haryana where he devised this whole thing. Kritika just graduated in December, and she focused more on the endothelial cells, the cells that line the blood vessel, and how those interact with the immune system, and built an intracellular signaling model. She first did this with the in vitro data, so cell culture data. So we have one paper that's in revisions related to this. We use logic-based ODEs, and so this has some gated signaling, but represents the logic with a sigmoidal hill function rather than just on and off. So it's not just discrete jumps, it has this continuous sigmoidal function to it. I won't go into all of those details. We also expanded the work using some natural language processing text mining work. And so I want to give a shout out to Cassie Mitchell's lab and her student Jennifer Dung, who really helped us be able to do this text mining of PubMed articles to enhance our search of which networks and which processes, both genes and proteins, that were really involved in the signaling network beyond the curated version that Krutika had just by manually reading papers. And then most recently, Krutika extended this work to in vivo, where we thought about how the spacing between these cells changes in disease, and been able to predict in vivo responses to different glucose dynamics in diabetic mice. So this is a separate preprint that's also under review now. And she explored in silico effects on these two main tissue outcomes. What's the spacing between the gaps between these endothelial cells, and how big those gaps are, and looked at different kind of treatment options and what would be the outcomes from that in diabetic mice. For the sake of time, I'll skip over this, but we have lots of ways in silico that we can think through potential therapeutics. And then we're working on now trying to combine some of these submodels in Nick's work. So using a finite element, this is showing things in FEBio. So we can couple fluid pressure, force balances, we can couple mass transport, and then put in the reaction rules from our other models into the health or dysfunction of different podocytes or endothelial or the matrix, and change material properties and see how that influences the protein excretion. So we're working on putting these together, but that's kind of toward the end goal here. So I've told you today about different tissues. I haven't told you about our bone and cancer work, but I did highlight some applications from lung and kidney and eye. I showcased work that uses agent-based modeling, some work that uses different kind of ODEs and PDEs and logic-based differential equations. In the eye work, I showed you several things that use finite element modeling to solve PDEs as well. So with that, I'd be happy to take any questions you might have and open up the floor for discussion.